Oh, it's sticking up. I think that's a good choice.
The conference call has been reconnected.
Conference.
Oh, welcome back.
Sorry to not know what you were asking for, Scott.
No problem. I was persistent.
So Chris Ma was able to identify that interesting sea cucumber. Yeah. Olafinius. With a GH. Olaf. GH in English. It's always a... All right. Now, while it's out there, she just hoover. When will we be back at this wall? Actually, this wall doesn't seem like it'll exist, right?
Alan, so you're about to collect the blue sponge, is that correct?
Yes. Is there anything else nearby there that... While we're at it?
I don't know if you earlier in the dive saw any of the yellow fans, but I noted there was a yellow fan above it. So just to get a look at it to see if we can get it to genus or something. Thanks.
Absolutely. See if that's Paramuricea or...
I think we only have about a half hour left. Just a heads up.
Yeah, Paramuricea Canthagorgia.
Mm-hmm.
Yeah, it's right above the sponge. You don't have to move or anything.
Yay. I think all we're going to get is Piscilla sclerata.
For the blue one?
Yeah. So the substrate is likely to break pretty easily.
Mm-hmm. You should be able to kind of peel them off the rock, and he's probably going to be very soft. Mm-hmm.
Yay! I'll take care of the worm.
Please take care of the worm. I can't even stand looking at it in the camera. It like freaks me out.
It's going to be so great.
Ugh. Be careful though. I don't want you to get stuck because sometimes they're really venomous. Poisonous.
There it
Ooh,
is!
there he
Yay! Success!
So we think that this has never been collected before. Is that the thought process?
Yeah.
Awesome. Did he get in?
Which did he go into?
Bucket 2.
No, he went... Oh, shoot.
Oh, and does... Oh, okay.
Yeah, I put the wrong thing.
That was bucket 2?
Yeah.
No, but can we get a good look at that yellow coral that's right in front of us? Yeah, no worries. That's good.
Oh, it is so great. Sorry, that was me looking at the scale worm inside the canister.
There's lots of ilex squid, huh? We think we're going to put that annelid in formalin?
Yes.
Just let the sample data managers know?
Mm-hmm.
So the last sampling we did of annelid, or the way I was taught in grad school, which was not that soon ago, was to cut the head off, put the head in formalin, and then put the rear end in ethanol for DNA. So we do use the rear end for DNA in the face because a lot of the identifying features are in those things on the head side. Whatever they are. The tentacly things on the front.
Yes, exactly.
That's cool.
Ooh, this is a nice...
Gorgonian.
Yeah, from this distance, this looks like it's probably in the Paramurcia group.
Paramurcia.
And not Acanthogorgia, but stand by on the zoom here.
Okay. He's very fuzzy looking. Like maybe
spiky.
Spiky,
Yeah.
spiky.
Yeah, that stickiness, actually maybe this is a Acanthogorgia. It's got a clear anthostele. That lower part, it almost looks like a cup that the tentacles arise from. They will attract into that, but the Acanthogorgiids have a very thin skeleton. Yeah, now I'm thinking this is in the family Acanthogorgiidae.
Awesome, thank you.
Cool.
We're good, guys. Thanks.
Really nice. Thanks again for that collect, if we didn't mention it.
Another little shrimp there. 9 o'clock. Maybe another bryozoan, a little fan. Little mesh fan.
Yeah.
Yep. Maybe not. Oh, yeah. Bryozoan, yeah.
Yeah, that bryozoan's been really likes this place.
Mm-hmm. Oh, Stichopathes. I think that's the first one we've seen. Yep. Still pretty sparse, though. I think maybe Chrysogorgid at 6 o'clock. Yep, there you go, right there. Oh no, maybe not. Yeah. Pink fan.
Yeah.
Yeah, bamboo coral. You can see the nodal branching. That's neat. It looks like maybe another bamboo coral right above it, or around 2 o'clock. And then we have that black coral, the whip, coming in at noon. This wall is still really steep here. Nice. So that's a really orange Stichopathes type of black coral.
It's just really bright.
Mm-hmm. Looks really healthy too. He's totally not sloughing off any tissue.
So the very thin coiling whip in front of it.
Yes.
Does that have octocoral polyps on it? The white one?
I'm not sure. Guys, can we swing? Yep, can we look at that white whip that's to the left? It's very tiny. Yep, right above the left laser. Yes, it does have polyps on the tip. Looks like it could be a bamboo coral, but it looks like the nodes are almost gold. Right? They're not really that
They're
brown.
really, really small space between those nodes.
Yep. Yeah, definitely bamboo, and I see those golden nodes. That's kind of interesting. Well, that's good. It's also really short internodes by the looks of things.
Mm-hmm.
An odd one. Thank you.
Yep. Then it looks like we have another bamboo coral at 3. This little rock here looks like it's about to fall.
Oh,
right there.
It does.
Give it a good kick.
Don't fly under that.
Yep. We've got a good bamboo coral, I think.
Yeah.
I would like to keep moving.
There you go.
Very silty sand. Thrusters cause that.
It does.
Yeah, it's very fine, silty sediment. Oh my goodness.
It just keeps going up and up and up.
Yeah, we're still 900. The never-ending, like, Twilight Zone depth. It's neat. There's, like, a really black layer just right in the middle there. 10 minutes. Okay, thank you. Maybe Chrysogorgid. I think that's what that is. Yeah. Chrysogorgid, another blue sponge. A couple of yellow sponges and white sponges in there. I can see how loose this looks. Like this could just go at any minute, right? That really, the layering. Like I feel like I could put my hand in there and just pop that whole section of rock right off. Just
Can we bring it up and then
Yeah.
sort through the organisms?
Yeah, of course.
So it looks like the ledge where we're seeing things is darker in color than what's around it. So I wonder if it's either a different mineral or it's been manganese encrusted, which is making it a little bit more stable. That's why there's some fauna growing out there. Says without knowing any geology.
What is that?
That certainly makes sense.
What is that? A siphonophore.
Oh, it is a siphonophore
That's
drifting
huge.
towards us. A long one.
Whoop, not anymore.
It said, I don't like what's near me.
Yeah, a cloud of schmutz floating around.
That was really cool.
If you run across any Chrysogorgia that have squat lobsters on them, that would be interesting to me.
Yeah, sure. We just saw a Chrysogorgid, so maybe we can swing back and look at it.
Wow, and its back stretched out.
That's neat.
Great example of how these guys... Whoa!
He's a couple meters long.
And then he's back together. Whoa,
Squid
and the
coming out.
squid. So many.
It's like a pogo stick.
You asked and you received. So it seems like it's done that 3 or 4 times. Oh, there's a nice close-up of its stem and all the different parts, all the different zooids. And those yellow parts, I believe those are the tentili that it's...
Ooh.
Whoa.
How it curls.
Yeah.
Very springy.
Yeah, I think that footage is good enough for someone to identify this species.
You can see the bells moving at the
Oh,
top.
there you can see the tentili. They're dangling from the tentacles, the little smaller yellow bits. So the larger yellow bits are most likely the gastrozoids, the feeding zooids.
Alan, I think I noticed 1 or 2 of those zooids was really large, and I remember a paper from Steve Haddock's lab that showed that the feeding zooids could ingest larvae. I think it was 3 to 5 times the size of the polyp, and they just expanded in size to accommodate it.
Cool.
I wonder if that's what we were looking at there.
Good chance.
Yeah, can we get back on that Chrysogorgid?
As the squid patrol goes by again.
This cloud of dust. It should clear out soon, but yeah. All right, Mary, we're getting close to zooming in on it. You've got a bit of a delay on your end on the video, so just give it a sec. We should be zooming in a little bit. There's also another squat lobster
on the rock.
At 8 o'clock on the rock.
That's the right squat lobster. Can you nab him?
Okay, you want us to grab one of these squat lobsters? Can we collect this Chrysogorgid?
Yes, please.
I'm not sure we have time, Mary.
Well, if you see one in the future, please grab it.
You're interested in the one specifically on the Chrysogorgids?
Yes, please.
All right. We've seen a few of these this cruise, so I'm sure we'll see more. But if we see one again, I will definitely grab it for you. Actually, we collected one, didn't we?
I know that we got a Paramaricia with one. No. That was with the Astroschema. Yeah, I think we did.
We got a Chrysogorgid.
Maybe right at the very beginning.
Hey, Mary, I can send you a picture of the one that I think we collected earlier in the cruise if you want. But we can still get another one if I'm wrong. Just that we're really ending up close to the end of this dive, and so just have like 3 or 4 minutes left.
Yeah, I'd like to see the picture.
Yeah, I'll get them to you. Do that after we're done sampling today. I'll email it to you.
There's another squat on the rock. Next.
Yeah, it's a
next
different
to this
squat
bamboo.
lobster. He's very armored.
So
Go ahead, Scott.
Are you able to get a little closer? Yeah, it looks like there's a couple of very weird places. You see those weird twisty branches just went off the top. There's another one there. I'll bet there's an anemone right in the center. We've been seeing this when anemones settle on these corals. They then cause subsequent growth of branches to be very aberrant relative to the rest of the colony.
Huh.
Odd.
The uppermost one, if we can get a zoom on. If you have time, I realize we're very close to the end and want to move, so don't worry about it.
Yeah, if you can zoom right
That
there.
squat lobster is Immunodopsis.
Immunodopsis.
Not a chirostylid.
Thank you.
Now look at that branch.
Fantastic zoom. There's definitely something in the center there. You can see there's a pinky blob.
Yeah. At like 3.
I wonder if it's the backside of an anemone. So the reason I'm really excited to see this observation, we were seeing a lot of that, especially this summer, and we've seen some in the Pacific, and we're starting to accumulate enough observations to be able to determine, is this only happening in one species of bamboo with one species of anemone, or is it a more broad-scale phenomena? Great. Thank you so much.
Yeah.
It's like the presence of the anemone sends the coral haywire, and it starts putting out branches in various directions, and it creates kind of a little cage around the anemone almost.
Huh. Okay, that's cool. You spot an amphipod in there? Yeah. On the branch to the right.
All right, we're good whenever you guys are.
The alternative, if that wasn't an anemone, I suppose it could be like an ascothoracican under the tissue. So kind of essentially like a relative of a barnacle parasite.
It looked pretty soft. I would have called it the foot of an anemone, just that the anemone's tentacles were tucked or not facing me. It looked like that, like little squished, retracted guy.
I agree, Stephanie. I think that's what it is, and that would certainly make sense with our previous observation.
Whoa.
Could this be the fabled top of a wall?
I don't think so.
No.
That I've often heard of but never seen?
We have another 70 meters to go before we hit the, right? Was it 8:30 or something?
No.
8:20. Yeah, forget it.
Yeah. So as far as we know, based on our data, tops of walls do not exist.
Yes, correct. Or we are just incapable of getting to them. They're like in the fifth dimension or something.
Good thing the walls themselves are so interesting.
I know, right?
They are.
Yes, please. The last Niskin bottle would be great. That's going to be sample 7.
Thank you very much.
Okay, thanks.
Thank you so much.
What number was that? That was 7W.
7W,
Okay.
yeah.
So we're ending this dive at roughly 892 meters. We started this dive. Oh, so okay. First, we're 30 miles south of the Dry Tortugas. And if you'd been following the cruise but didn't tune in yet today, we had to come off the Blake Plateau the past 2 days and escape some gnarly weather. So we've come down to the south of the Keys. So we're diving right now in Tortugas Valley. In 1,000 meters of water is about where we landed, and the plan was, based on the multi-beam, to go up a really tall wall. We didn't really find that wall until, I don't know. It feels like 20 minutes ago. It's probably about a couple hours in. It took us a while to find the wall, which is...
Oh, many hours in.
Yeah, it was many hours in. So the wall wasn't where we were expecting it to be. So the thought process is that it sloughed off between the time the multi-beam was collected roughly in 2014 until today. So I think the plan is tonight to just run a multi-beam over it and see how it's changed. So because it looks like it just fell. The whole thing just avalanched down. Because we were supposed to get to 820 at the top of the wall, but we were at where we ended, 9-something, halfway up the wall. Yeah. And we should have been at the top of the wall hours ago. So it looks like a big chunk of it fell off.
I know.
Yeah.
Which is not surprising when you see how we could see its, small slides. It's not very consolidated.
Yeah.
So
It looked like that soft clay, or really not quite solidified hard bottom. It was more of that clay, malleable almost.
Yeah.
We had picked something similar up in the sinkhole on the far east of the Blake Plateau. What day was that? Friday.
Mm-hmm.
So when we got down, it was mostly sand, hardly any waves. It was mostly bioturbated sand. So the so that means there's fauna or stuff growing inside the sediment or causing divots and mounds to form.
We saw a lot of mobile fauna on top of that sand, so especially this ilex squid,
really
which was with us from the beginning of the dive all the way to the end,
Yeah, absolutely.
as well as many different kinds of fish.
Yeah, we had a lot of fish today,
cuspials,
too.
and some really great tripod fish. 2 different species of tripod fish.
And then when we finally reached the wall, we did find some corals and some our typical, hard bottom fauna that we see at this depth.
Encrusting demo sponges.
We collected 5 water samples, 1 at 500, 1 at 1,000, 1 at the end of the dive, and then 2 in the middle. We also collected this really
beautiful scale worm.
Oh, they freak me out so bad.
Not everyone agrees.
Yeah, I know. And then number 6 was, the blue sponge.
Yes,
Sample 6
Pisciclesclerid sponge. At least that's the hypothesis.
Yep, so it's a really cool blue sponge. It was actually really common on the hard bottom. We see quite a few of them, and I think we've actually seen them most of this cruise. They're really tiny little guys. They're, 5-6 centimeters long, but they're not uncommon, on the hard areas. What else do we get? Lots of bamboo corals. I saw 1 Stichopathes, and I think that was the only. Oh no, maybe 1 other black coral.
Parantopathes.
And
we saw
Bathypathes.
Bathypathes for sure.
Bathypathes. What else do we got?
I'll also throw out that potential acanthogorgid or placagorgia, and I just want to note that the bamboo coral whip that we zoomed in on that had those golden nodes and really short internodes, that was something really unusual, and so I'm going to keep an eye out to see if we are lucky enough to encounter that again. I'm certain we won't, but something very odd. So that was a great sighting. And then that last coral we just looked at with the odd branching because of the putative anemone, also really good observation.
Okay, awesome.
That's great.
Then we had a really cool ophiuroid that Chris Ma was really excited about that looked like he had a little red hat on.
Yes.
But we lost it, but it was really tiny.
That's only because it sat down in the sediment right next to the squat lobster we were looking at. We also saw, we got some nice siphonophore sightings.
Oh, yeah,
we had the
dandelion.
benthic siphonophore, the dandelion siphonophore, and then also this Physonex siphonophore at the end that was sort of
they're really long,
repeatedly stretching itself out and then contracting, and we got a really nice view of many of its zooids.
We had Chasion with the red deep sea crab. We saw a red deep sea crab eating an illic squid. That was cool.
Yeah.
What else?
And I know you're saving for best the sea pens.
Oh yeah, of course, the sea pens.
Of course,
multiple sea pens in the sediment. I think that's about it. Oh, a couple pycnogonids or sea spiders.
Pneumaticaris shrimp were
that's right, some shrimp.
Pretty common.
That asteroid.
Water walking isopod.
Yes, we did see that.
And I'm sad to say that you have forgotten that the devilfish.
Oh yeah. Oh my God, how did I forget him?
The octopod.
The octopus. Yeah, super cool.
Very, very cool.
Yeah. So I think that about sums it up. So I think, what's the plan for tomorrow? To go towards the west? Yeah, and we're going to have a post-dive call shortly. So just hang on the line if you want to be involved in our planning call for tomorrow. We're going to have that right after this. So either dial in or just hang out. We'll be with you shortly because we're going to be going to the west, probably south of Pulley Ridge. Not to Pulley Ridge. Pulley Ridge is only 60 meters, so we're definitely not diving Pulley Ridge, but south of it on the escarpment. And, tune in tomorrow. Hopefully we've got good enough weather to dive again. Yeah. It looks nice out here. So, beautiful. Keys are gorgeous.
Yeah, and actually now that we rehashed all that, even though it was a sand dive and they were sort of anxious to get to the walls, there was a lot to see.
Yeah, there was a lot.
And there was a really big diversity of things that were exciting.
Yeah, it was a good dive. That's always a good dive. Any day diving's better than any day in the office. So. No offense, all my landside people.
True.
I know you're in the office watching, but.
Yeah.
All right. We will see you all tomorrow. Have a wonderful
evening. Looking forward to it.